Hello students, this is Mrs. Sonal Patil. In a previous class we have studied about translation and we had started with the regulation of gene expression. Today we will continue with that. Regulation of gene expression and most of the vocabulary used have come from the studies in the colon bacterium E. coli by French molecular geneticist Jacob and Monod. They proposed a model called operon model to explain the genetic basis of enzyme induction and repression. Proteins are the building blocks of an organism synthesized in a cell as they are directed by DNA. The protein molecules are produced when the cell requires and they are not produced when there is no necessary of proteins. Hence, DNA undergoes switch on and switch off mechanism for protein synthesis is called differential gene action. It was explained by Jacob and Monod under Lac-Operon concept that explains the regulation of gene action. As I told, they conducted the experiments on the utilization of lactose by E. coli bacteria. When the lactose molecules are absent in the nutrient medium, the DNA of bacteria is switched off and do not produce enzymes, that is proteins. When the lactose molecules are present, the DNA of bacteria is switched on to produce enzymes which are necessary to break lactose into glucose and galactose. The DNA of bacteria consists of several genes arranged in a particular manner. They are regulator gene, promoter gene, operator gene, and structural genes, that is Z, Y, and A. Here, the operator gene, it is a gene which directly regulates the formation of mRNA of the structural gene. The operator gene acts as a switch, either blocking the structural genes or permitting them to function. Coming to the regulator gene, the gene which regulates the action of an operator gene is called a regulatory gene. It controls the activity of an operator gene by producing inhibitors of gene activity called repressors or suppressors. The repressor is a protein which causes repression. Repression is nothing but it is a phenomenon in which the synthesis of a particular protein or enzyme is repressed or stopped. Inducer. Now here the word inducer is been used. Inducer, it is a chemical which inactivates the repressor by modifying their structure. Lactose is the inducer in Lac-Operon of E. coli. Promoter gene, it lies between the regulator gene and the operator gene. The promoter gene is a region on mRNA for binding of enzyme DNA dependent RNA polymerase which is responsible for transcription. The enzyme RNA polymerase always binds to the promoter region by identifying the specific nucleotide sequences TATAAT. This region is called Tata box or it is called as Pribno box. So the cluster of structural genes together with the operator gene is called operon genes. When RNA polymerase enzyme transcribes the structural genes that produce 3 mRNA strands, these mRNA strands undergo translation to produce 3 enzymes such as Z gene produce beta galactosidase, Y gene produces beta galactosidase permease and A gene produces beta galactoside transacetylase. These 3 enzymes together break lactose into glucose and galactose. This is a Lac-Operon been shown here with the promoter gene, okay, operator, and there are structural genes. switch off mechanism. When lactose molecules are absent in the nutrient medium, DNA is switched off for protein synthesis. So when I told lactose is acting as an inducer, when lactose is absent in the nutrient medium, DNA is switched off for protein synthesis. Means what? The regulator gene produce a kind of protein called repressor that binds to the operator. This do not allow RNA polymerase enzyme to transcribe the structural genes. Since the transcription of structural genes is not occurring, then the required proteins are not synthesized. Hence, there is no transcription, translation, and the required protein is not synthesized. So, this mechanism is called as switch off mechanism. Very important. This mechanism will be asked in the exam. Lack operon concept is important for 5 marks. Switch on mechanism. When the lactose molecules are present in the nutrient medium, DNA is switched on for protein synthesis. The lactose molecules added in the nutrient medium acts as inducer, which binds to the repressor to form inducer-repressor complex. Now, the repressor become inactive and release from the operator gene. This enables the RNA polymerase enzyme to transcribe all the 3 structural genes, that is Z, Y and A. This results in the synthesis of 3 mRNA molecules continuously are called polycistronic mRNA. All these 3 mRNA strands undergo translation to produce 3 polypeptide chains which act as 3 enzymes such as beta-galactosidase permease and beta-galactosidase galactoside transacetylase. These 3 enzymes together break lactose into glucose and galactose. In polycistronic mRNA, the product of first gene activates the second gene, the product of second gene activates the third gene, and so on. This is called pre-programmed circuits of gene action. So here this has been explained, the switch on and the switch off mechanism. Here again they have shown it diagrammatically, seen in the absence of inducer what is happening, and in the presence of inducer how the enzymes are synthesized. So here we can see in the absence of inducer what is happening. When there is no lactose in the culture or in the medium, the regulator gene produces mRNA. Okay, regulator gene produces mRNA, which in turn produces repressor protein or suppressor protein. The repressor protein, when binds to the operator gene, blocks it, results in the inactivation of the structural gene so that the transcription and protein synthesis is stopped. Now the structural genes are said to be switched off. So here you just see regulator gene in the absence of the inducer. Okay, it will produce mRNA. mRNA in turn produces repressor protein. This repressor protein will bind to the operator and then it will block the that is, it results in the inactivation of the structural genes are inactivated, and because of which the transcription is not occurring and the protein synthesis is stopped. So this mechanism is called as switch off mechanism. Now when there is lactose in the medium, a few molecules of lactose are carried on into the bacterial cell and they bind to the repressor protein. They bind to the repressor protein produced by the regulator gene to form inducer-repressor complex. So inducer-repressor complex is formed. It results in the inactivation of repressor protein, and that fails to bind to the inactive repressor is formed. It will fail to bind. It will not bind to the operator gene. Then operator gene signals the structural genes to produce mRNA, which in turn produces corresponding proteins which function as enzymes. Among the 3 enzymes, beta-galactosidase catalyze the conversion of lactose sugar into glucose and galactose. Since the structural genes are now acting, they are said to be on switched on condition. The promoter gene is a site for binding of enzyme DNA dependent RNA polymerase, which acts on the structural genes and controls transcription. So that was a lac operon concept, important question for 5 marks. Coming to the next, Human Genome Project. The term genome was coined by H. Winkler to the complete set of chromosomal and extrachromosomal genes of an organism or a virus. We are aware that each human individual has a separate identity which differentiates him from other, and his identity is due to the presence of specific gene makeup. So if two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. The Human Genome Project was a mega project. Aims of the project are as follows. Human genome is said to have approximately 3 into 109 base pairs, and if the cost of sequencing requires is US, that is $3 per base pair, the total estimated cost of the project would be approximately 9 billion US dollars. Further, if the obtained sequences were to be stored in a typed form or in books, and if each page of the book contained 1000 letters and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high speed computational devices for data storage and retrieval and analysis. Human Genome Project was closely associated with the rapid development of new area in biology called bioinformatics. Goals of Human Genome Project. Some of the important goals of Human Genome Project are as follows. Identify all the approximately 20,000 to 25,000 genes in human DNA. Determine the sequences of 3 billion chemical base pairs that make up human DNA. Store this information in databases. Improve tools for data analysis. Transfer related technologies to other sections such as industries. Address the ethical, legal and social issues that may arise from the project. So these are the goals of the Human Genome Project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of Human Genome Project, the Wellcome Trust became a major partner. Additional contributions came from Japan, France, Germany, China and others. The project was completed in 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat and someday prevent the thousands of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Drosophila, etc., plants and all have been sequenced. So this was about the Human Genome Project. We have studied about the goals. We'll stop here. In the next class, we will study about the methodologies involved in the Human Genome Project, and we will end up with DNA fingerprinting. That will end the chapter, that is molecular basis of inheritance. For today, we'll stop here. Thank you.